Welcome, curious minds, to the deep dive. When we first learned about genetics, it probably felt so, well, neat.
Right.
Think back to Mendel and those iconic pea plants. Tall or short, green or yellow. Simple, dominant, and recessive, leading to those perfectly predictable 3.1 ratios. It's almost too tidy.
Yeah, it sets an expectation that isn't always met.
But here's where it gets really fascinating. Biology, as it turns out, is rarely that simple. And that thrilling complexity is exactly what we're here to unpack today.
You got it. Here on the deep dive, our mission is really to cut through all that information overload, giving you that fast track to being truly well-informed. Today, we're diving into the captivating world of extensions of Mendelian inheritance.
That's right. We're extracting our insights directly from Chapter 4 of Robert J. Brooker's Genetics, Analysis and Principles, 7th edition. Our goal today is to unravel how traits are influenced by factors far beyond those straightforward dominant-tracive relationships.
We'll explore the subtle underlying molecular mechanisms, see how these complexities play out in the real world, from the color of flowers to challenging human diseases.
And arm you with the key terminology and experimental methods that geneticists use every day.
Indeed. We'll explore why some traits seem to skip generations entirely, how the environment actively shapes gene expression in surprising ways.
And even how multiple genes can interact in a kind of grand genetic symphony to create outcomes Mendel never dreamed of.
It's a journey that truly reveals the elegance and the nuance of genetic inheritance.
So let's begin by just grounding ourselves in Mendel's foundational laws. Segregation and independent assortment. These are the bedrock, right? Teaching us how allele is separate and how different genes sort.
Absolutely fundamental.
For what we call simple Mendelian inheritance, one allele strictly dominates another, reliably leading to those classic 3.1 phenotypic ratios we all learned about.
And while Mendel's work was absolutely groundbreaking, truly revolutionary, what geneticists have discovered since is this huge diversity of mechanisms by which alleles actually affect traits.
So it doesn't mean Mendel was wrong.
Not at all. It just means his work was the crucial starting point for understanding a much more intricate biological reality. The vast majority of traits we observe in nature simply don't produce those classic 3.1 ratios.
So understanding these more complex patterns isn't just academic, it's vital for everything from agriculture to medicine. Forget those simple 3.1 ratios. Most traits, really, from disease susceptibility to even subtle variations in appearance, are shaped by layers of interaction.
Exactly. And our two main goals today are, first, unlocking how to predict the outcomes of genetic crosses that seem to defy that simple math.
And maybe more importantly, deciphering the intricate molecular journey from DNA code from the gene itself to the visible traits we actually observe, the phenotype.
Precisely. Broadly speaking, the factors that influence how a trait is expressed beyond simple dominance includes things like the specific level of protein expression. So, the sex of the individual, the presence of multiple alleles for a single gene within a population, the profound effects of the environment.
Right.
And, perhaps most fascinating, the intricate interactions between different genes, often many genes working together.
All right, let's really unpack those different ways alleles can interact them, because it clearly goes way beyond just dominant and recessive.
Okay, so one of the most intriguing twists we see is when a dominant allele is clearly present in an individual's genotype, but the expected phenotype just isn't expressed. We call this incomplete penetrance.
Incomplete penetrance.
And it's measured at the population level. So if a dominant allele is present in, say, 100 people, but only 60 of them actually show the trait, we'd say that allele is 60% penetrant in that population.
So it's like the gene could act, but sometimes doesn't.
Exactly. Something is holding it back or preventing it from fully manifesting in some individuals.
That must make understanding family pedigrees incredibly tricky.
It certainly can. The classic real-world example here is polydactyly, the dominant trait that typically causes extra fingers or toes.
Right, I've heard of that.
In some family trees, you might see an individual who clearly carries the polydactyly allele we know, because they pass it on to their children, but they don't actually show the trait themselves. They have a normal number of digits.
So the gene is there, but nothing.
Right. It's as if other genetic factors or maybe environmental influences are acting like dimmer switches, influencing whether the trait appears at all.
Okay, so what's going on at the molecular level there?
Well, it could be environmental influences, as I mentioned, or even modifier genes. Other genes that are actively counteracting the dominant allele's effects, preventing its full expression.
Got it.
And while we're on the subject, it's important to distinguish this from expressivity. That's a related concept, but different.
Okay, expressivity, how does that differ?
Expressivity describes the degree to which a trait is expressed when it is penetrant. So, sticking with polydactyly, one person with the allele might have just a tiny extra nubbin of a toe, while another person with the same allele might have multiple fully formed extra digits on both hands and feet. The gene is penetrant in both cases, but the expressivity, the intensity of the phenotype, varies widely.
Okay, so incomplete penetrance is if it shows up, and expressivity is how much it shows up.
You got it. But what happens when it's not a question of if or how much, but a completely different manifestation, like a middle ground?
A blend.
Exactly. That's where incomplete dominance comes in. In this scenario, the heterozygote has a phenotype that is genuinely intermediate between the two homozygotes. It's not one or the other, but a distinct third phenotype, a blend.
The 4 o'clock plant provides that vivid illustration, doesn't it? Yeah. If you cross a homozygous red-flowered plant, let's say CRCR, with a homozygous white-flowered plant, CWCW, their F1 offspring aren't red or white, they're all pink, CRCW.
A true intermediate. And then, if you self-fertilize those F1 pink flowers,
the F2 generation gives you that characteristic 1.2.1 phenotypic ratio. 1 red, 2 pink, 1 white. It's a clear deviation from Mendel's 3.1.
And from a molecular standpoint, this often happens because the single functional allele in the heterozygote only produces about half the amount of the necessary enzyme or protein.
Ah, so 50% isn't enough.
Right. That 50% dose just isn't sufficient to reach the critical threshold for the full-strength trait seen in the dominant homozygote, leading to that intermediate phenotype. Like half the red pigment makes pink, not red.
Okay, that makes sense.
And what's truly fascinating here is how our very definition of dominance can shift depending on how closely we're looking.
What do you mean?
Well, take pea seed shape again.
Right.
Visually, a pea that's homozygous dominant for round, RR, looks just as round as a heterozygote. To our eyes, R looks completely dominant. But if you dissect those seeds and look under a microscope, analyzing the starch granules, you'd find that the heterozygous R seeds actually have an intermediate amount and form of starch compared to the R homozygotes.
Wow. So at the molecular level...
At the molecular level of starch biosynthesis, it's actually incomplete dominance, even if it looks like simple dominance to the naked eye.
It's a powerful reminder that biology's truths are often revealed in the details.
Absolutely. Which brings us nicely to overdominance, sometimes called heterozygote advantage.
Ah, yes. Where the heterozygote actually has greater reproductive success or fitness than either homozygote. This isn't about an intermediate phenotype, but a genuinely superior one in some context.
Precisely. And the most powerful human case study here is sickle cell disease.
Right.
Individuals who are homozygous for the HBS allele, HBS, develop severe sickle cell disease. Their red blood cells deform into that sickle shape, causing blockages, pain, organ damage. It's a devastating condition.
But here's the incredible twist. The heterozygotes, those with one normal HBA allele and one sickle cell HBS allele, HBA-HBS, are largely unaffected by sickle cell disease, and remarkably, they are also resistant to malaria.
That's the key. Their red blood cells, with that mix of hemoglobin types, tend to rupture when infected by the malaria parasite, which prevents the parasite from multiplying effectively.
So in regions where malaria is prevalent, these heterozygotes have a significant survival advantage over both the HBA-HBA individuals who are susceptible to malaria and the HBH-HBS individuals who suffer from sickle cell disease.
Exactly. And this survival advantage explains why the HBS allele persists at relatively high frequencies in those populations, despite its severe effects in homozygotes. Evolution finding a balance.
It's quite something, isn't it? The way evolution can even co-opt what seems like a detrimental allele and turn it into an advantage in a specific environment.
It really is. And thinking more broadly, overdominance can arise from a few molecular explanations. Increased disease resistance is one, like with sickle cell, and some speculate it might play a role for PKU and Tay-Sachs heterozygotes too.
Okay.
Another possibility is enhanced protein function. Maybe a protein formed from two different subunits coded by the two alleles, a heterodimer, works better or is more stable than one made from identical subunits, a homodimer.
Interesting.
Or it could be that the proteins produced by each allele function optimally under slightly different conditions, say different temperatures or pH levels. So the heterozygote has an advantage across a wider range of environmental factors.
That's remarkable flexibility. Now, let's switch gears slightly to codominance. How does that differ from incomplete dominance where we saw the blend?
Good question. With codominance, the heterozygote expresses both alleles simultaneously and distinctly. There's no intermediate phenotype. Instead, you see both traits fully expressed side by side.
The classic example, the one most people know, is human ABO blood types, right?
Exactly. If you inherit both the IA allele and the IB allele, you don't get some kind of AB blend blood type. You get type AB blood.
Meaning your red blood cells display both the A antigens and the B antigens, clearly and completely on their surface.
Precisely. And the molecular basis is quite elegant. The I allele, for type O, produces an inactive enzyme. It doesn't add the final specific sugar to the precursor molecule on the cell surface.
So that's the base state type O?
Basically, yes. It results in what's called the H antigen. Now, the IA and IB alleles encode functional glycosyltransferase enzymes, but they have subtly different active sites.
Different tools for the job?
You could say that. The IA enzyme attaches a specific sugar called N-acetylgalactosamine, creating the A antigen. The IB enzyme attaches a different sugar, galactose, creating the B antigen.
So if you have both IA and IB.
You make both types of functional enzymes. Your cells attach both N-acetylgalactosamine and galactose to the precursor, resulting in both A and B antigens being fully expressed on the cell surface.
Predominant. And this, of course, has critical real-world applications, especially for blood transfusions. Understanding codominance is absolutely vital for ensuring compatibility and avoiding those dangerous agglutination or clumping reactions.
Definitely. It's why type AB individuals are called universal recipients. They already have both A and B antigens, and type O are universal donors, lacking the A and B antigens that could trigger a reaction.
So far, we've seen how individual alleles can interact in surprising ways. But what if the genetic context itself changes based on sex? How does being male or female reshape how genes are expressed?
That's a crucial area. First, we need to consider genes located directly on the sex chromosomes, particularly the X chromosome. This is X-linked inheritance.
Genes on the X, but not usually the Y.
Primarily, yes. And because males typically have only one X chromosome, XY, they are homozygous for genes on the X. They only have one copy.
Which means
They're much more susceptible to X-linked recessive disorders. If they inherit just one copy of the recessive allele on their single X, they'll express the trait.
Whereas females, XX, have two X chromosomes, so they'd usually need two copies of the recessive allele to show the trait.
Exactly. They can be carriers with one recessive allele, but often don't show the phenotype. This makes X-linked recessive conditions far more common in males.
Duchenne muscular dystrophy, DMD, is a stark illustration, right? A severe X-linked recessive disorder causing muscle degeneration, primarily seen in boys.
Yes, a classic example. Affected males usually inherit the allele from their carrier mothers. Females are very rarely affected.
And experimentally, reciprocal crosses can help identify X-linked traits, can't they?
They're a key tool. If you swap the sexes of the parents with specific phenotypes in a cross, and you get different results in the F1 generation depending on which way you did the cross, that's a strong indicator of X-linked inheritance.
Okay. What about the Y chromosome?
Well, there are Y-linked or holandric genes. These are found only on the Y chromosome and are passed directly from father to son. The SRY gene, critical for male development, is a prime example.
And wasn't there something about genes on both X and Y?
Yes, pseudo-autosomal inheritance. There are small regions at the tips of the X and Y chromosomes that are homologous. They carry the same genes, like the MYC2 gene.
So they can pair up during meiosis.
Precisely. And genes in these pseudo-autosomal regions are inherited more like autosomal genes, since both males and females effectively have two copies.
Okay, so that's genes on the sex chromosomes, but sex can influence genes elsewhere too.
Absolutely. Take sex-influenced inheritance. This is where an allele's dominance differs between the sexes. The same allele might be dominant in males but recessive in females, or vice versa.
Even if the gene is on an autosome.
Yes, typically these are autosomal genes. The expression pattern is influenced by sex hormones.
The example of skers in cattle comes to mind, those horn-like growths.
A perfect example. The allele for skers, SAE, is dominant in males. A male with just one SC allele will have skers, but in females that same C allele is recessive. A female needs two copies, SCSC, to develop skers.
A complete reversal of dominance based on sex? Wow.
Then there's sex-limited inheritance. This is even more distinct. The trait literally occurs in only one of the two sexes, period. The genes might be present in both, but they're only expressed in one sex.
Often due to hormones or specific developmental pathways.
Exactly. Think about sexual dimorphism. The obvious physical differences between males and females of many species, like the elaborate plumage of a rooster compared to a hen.
Right, hens don't develop those large combs and bright feathers, even if they have the genes for them.
That's sex-limited expression. And of course, the most fundamental examples are the presence of ovaries in females and tests in males' traits strictly limited to one sex.
Okay, one more category in this section. Lethal alleles. Sounds ominous.
They can be. A lethal allele is one that has the potential to cause death. Most are recessive and often affect essential genes absolutely required for survival.
So if an individual inherits 2 copies of a recessive lethal allele,
It's usually fatal, often very early in development, maybe even before birth.
are there variations, like alleles that aren't always lethal?
Yes, definitely. We have conditional lethal alleles. These only cause death under specific environmental conditions. Think of those temperature-sensitive alleles in fruit flies. Larvae might die at high temperatures, but survive fine at cooler ones.
Or like the fava bean sensitivity in humans with G6PD deficiency.
Exactly. Eating fava beans can trigger a severe, potentially lethal reaction in susceptible individuals, but otherwise they might be fine. That's conditional lethality.
Okay.
And then there are semi-lethal alleles, which kill some individuals who inherit the critical genotype, but not all. The white-eyed allele in Drosophila is an example. It reduces viability, but some survive.
And how do these affect inheritance ratios?
They distort them, often predictably. The classic example is the Manx cat, known for its shortened or absent tail. This is caused by a dominant mutant allele. Let's call it M.
Okay.
Heterozygotes are Manx. But being homozygous for the dominant allele, is lethal early in embryonic development.
So if you cross 2 Manx cats, MS and MM,
You don't get the expected 1 millimeter, 2 millimeter, 1 millimeter ratio among the kittens born. The embryos don't survive. So the observed ratio among surviving offspring is 2 Manx, 1 normal tail. That characteristic 2.1 ratio is a strong clue for a recessive lethal allele masked by a dominant phenotype in the heterozygote, or in this case, a dominant lethal allele in homozygous state.
and the timing can vary too, right? Lethality isn't always embryonic.
Absolutely. The age of onset varies dramatically. It can be embryonic like the Manx allele, or cause death in childhood like Tay-Sachs disease, or even manifest much later in adulthood like Huntington disease, which typically doesn't show symptoms until middle age.
We've covered single gene effects, allele interactions, the influence of sex, but genetics gets even more complex when single genes have multiple jobs or when multiple genes team up.
Right. Let's dive into that. First, pleiotropy. This is when a single gene influences 2 or more seemingly unrelated phenotypic traits.
One gene, multiple effects.
Exactly. And contrary to the simple one gene, one trait idea we sometimes learned initially, pleiotropy is actually extremely common. Most genes probably have multiple effects.
Cystic fibrosis seems like a prime example in humans. One faulty gene, the CSTR gene,
Which codes for a chloride ion transporter.
leads to a whole suite of problems. Thick mucus in the lungs causing breathing issues and infections, blocked ducts in the pancreas leading to digestive problems, salty sweat.
All stemming from that single gene defect impacting chloride ion transport in different tissues.
Why does pleiotropy happen?
Several reasons. The gene's product might be fundamental to the function of many different cell types, or the gene might be expressed in different cell types throughout the body, or it could be expressed at different stages of development, impacting various processes over time.
It really highlights how interconnected biological systems are.
It does. And as we've touched on, the environment constantly interacts with these genetic factors. Remember the Arctic fox changing fur color with the seasons?
Right, a temperature-sensitive allele controlling pigment production.
Or PKU, where dietary intervention and environmental change can prevent the severe effects of the genetic mutation.
It underscores that phenotype is almost always genotype plus environment.
Precisely. And geneticists talk about the norm of reaction for a given genotype. That's the range of possible phenotypes that genotype can produce across different environmental conditions.
Like the fruit fly eye facets varying with temperature.
Exactly. The same genotype can result in different numbers of facets depending on the developmental temperature, showing its norm of reaction for that trait under those conditions.
Okay, so beyond pleiotropy and environment, what about when different genes interact to shape one trait?
Now we're getting into gene interactions. This is when allelic variants of 2 or more different genes collaborate or interfere to affect a single phenotype. This is how many complex traits are built.
Bateson and Punnett's sweet pea experiment is the go-to example here, isn't it? Crossing 2 different true-breeding white-flowered varieties.
And getting all purple F1 flowers, which was totally unexpected based on simple inheritance.
And then self-crossing those purple F1s yielded an F2 generation with a ratio of 9 purple to 7 white flowers. Not 3.1, not 1.2.1, but 9.7.
That 9.7 ratio is a classic signature of epistasis. Epistasis is when the alleles of one gene mask or hide the phenotypic effects of the alleles of another gene.
So one gene can effectively silence another?
In a way, yes. In the sweet peas, it turns out there's a biochemical pathway to make the purple pigment. Let's say you need enzyme C from gene C to convert a colorless precursor to an intermediate, and then enzyme P from gene P to convert the intermediate to purple pigment.
A two-step process.
Right. If a plant is homozygous recessive for either gene, CCPP, CCPP, or CCPP, 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 it can't complete the pathway. Being CC means no enzyme C, so you get stuck at the precursor. Being PP means no enzyme P, so you get stuck at the intermediate. Either way, no purple pigment, the flower's white.
So the homozygous recessive state of either gene is epistatic to the dominant allele of the other gene, masking the potential for purple.
Precisely. That's recessive epistasis, leading to the 9.7 ratio. Only plants with at least one dominant C allele and at least one dominant P allele, CP, can make purple pigment.
And that initial cross-white X-white giving purple F1 that illustrates complementation.
Beautifully. It tells you the two white parent plants had mutations in different genes. For example, one was CCPP, the other was CCPP. Each parent provided the functional allele the other was missing.
They complemented each other's defect.
Exactly, restoring the wild-type purple phenotype in the F1 CCPP.
Okay, epistasis is masking. Are there other kinds of gene interactions?
Yes, there's also the gene modifier effect. Here, an allele of one gene modifies or changes, but doesn't completely mask the phenotypic expression of another gene.
Like fine-tuning instead of silencing.
Kind of. Think about rodent coat color, often involving the agouti gene, AA, and the color gene, CC. The interaction can give a 9 agouti, 3 black colored, 4 albino ratio.
Okay, how does that work?
Well, the C gene is needed for any pigment production. If a rodent is CC, homozygous recessive, it's albino, regardless of the agouti gene. So CC is epistatic to AA.
Right, albinism masks everything else.
But if the rodent has at least one dominant C allele, allowing pigment, then the agouti gene comes into play. Normally, the dominant A allele gives the agouti pattern, yellow and black bands on each hair, but the homozygous recessive genotype modifies this, resulting in an all-black coat instead of agouti.
So A modifies the phenotype produced by C, changing it from agouti to black.
You've got it. It's modification within the context set by the C gene.
Fascinating. And one last interaction type.
Gene redundancy. This is where losing the function of a single gene has no noticeable effect on the phenotype, because another gene can compensate for its loss. It's like having a backup system.
Only when you lose both genes do you see a change.
Exactly. This is often discovered through gene knockout experiments, where researchers intentionally disable a gene. Sometimes knocking out one gene did surprisingly little, pointing towards redundancy.
Why does this happen? Are these related genes?
Often, yes. It can be due to gene duplication events in evolution, creating multiple copies of a gene, paralogs, that retain similar or overlapping functions. If one is lost, the other ace can often pick up the slack.
Or maybe different proteins involved in the same pathway?
That too. Proteins involved in parallel pathways or common cellular functions might be able to compensate if one is missing.
Is there a good plant example for this?
The shepherd's purr seed capsule shape is a classic. Normally the capsules are triangular, but an ovate, egg-like shape appears only when two different genes, let's call them T and V, are both homozygous recessive, TTV.
So having just one functional copy of either T or V is enough?
Correct. As long as there's at least one T allele or one V allele, TV, TTV, or TV, the capsule is triangular. This redundancy leads to a 15.1 ratio of triangular to ovate capsules in the F2 generation of appropriate crosses. 15 genotypes result in triangular. Only one TTVV gives ovate.
15 to 1. That really shows the power of redundancy. Wow, what an incredible journey through the sheer intricacies of genetic inheritance. It really takes you from Mendel's neat garden plot into this complex, dynamic landscape.
It certainly does. From the subtle dance of protein expression levels and incomplete dominance to the interplay of sex chromosomes and hormones.
All the way to the grand symphony of pleiotropy, epistasis, and multiple genes interacting with the environment. It's abundantly clear that while Mendel laid the foundational groundwork,
and absolutely crucial groundwork it was,
life's genetic tapestry is far richer, far more nuanced than he could possibly have imagined back then.
And this isn't just, abstract theory for geneticists. These extensions to Mendelian inheritance explain so much about the real world, about who we are, why organisms look and function the way they do, our varying susceptibility to certain traits and diseases.
It really changes how you think about inheritance.
It encourages us to always look deeper, I think, to question our assumptions about what appears to be a simple trait. Because very often when you scratch the surface, there's a lot more going on underneath.
So maybe we can leave you, our listeners, with this thought to ponder. Given this vast, interconnected network of gene interactions, modifier effects, environmental influences, everything we've talked about, how many traits that we currently consider simple might actually reveal hidden layers of complexity if we just knew where and perhaps how deeply to look?
That's a great question to mull over. It really speaks to the ongoing adventure of genetics. Thank you for joining us for this deep dive and being part of the Last Minute Lecture family.
Yes, thank you. We look forward to exploring more with you next time on The Deep Dive.